to introduce our webinar speaker, Dr. Ronny Schmidt. Ronny studied chemistry at the Technical University of Braunschweig and Heidelberg University between 2003 and 2008, followed by a doctorate in functional genomics under the instruction of Dr. Jörg Hoheisel, which he completed in 2011. From 2012 and 2013, Ronny joined the Barbraham Institute in Cambridge, UK as a postdoctoral scientist. He was a project manager for protein array technology at Cambridge Protein Arrays until 2014. Since August of 2014, Ronny heads business development and contract research at Psyomics in Heidelberg in Germany. So Ronny, the virtual floor is yours. Please take it away.
Thank you very much for the nice introduction, and hello everyone. So, Psyomics. The title says Proteomic Discovery to Clinical Application, and I hope in the next 30 minutes I will guide you through the process, explain our platform, and answer all your questions. First, Psyomics is a spin-off from the German Cancer Research Center, and we were founded in 2013, and we are a fast-growing spin-off. We have main development partners, having the DKFZ, so the German Cancer Research Center, but also major university clinics all over Europe, and some of them in the US. So what's our vision? Why have we founded Psyomics? So we want to advance precision medicine through innovative biomarker signatures. A few words, biomarker signatures. You all know there are plenty of candidates around, and we focus on proteomic discovery. And the reason behind this is the correlation between DNA, RNA, and the actual protein as the phenotypic representative of a disease, of a certain state, is not necessarily very good. So there was a large study conducted within the Human Protein Atlas project, and it showed a mean correlation of RNA to protein of about 0.5. So that's as good as a coin flip. And we think if you go for serious biomarker discovery with a low attrition rate and fast project timelines, you should really go for the phenotype directly, and therefore having the chance of a coin flip is just not good enough. So look at the protein level, not necessarily the RNA level. Usually, when you want to look at protein level, you have two major blocks of methods. So you have either the mass spectrometry or the classical immunoassay methods, like ELISA's speed-based assays, flow cytometry, for example. The problem with mass spec, you have a high content and a large hit list. So if you get 1,000 possible candidates, where do you start with your validation? I mean, it's an advantage on the one hand, but a serious disadvantage when you go for a follow-up. Also, the throughput in mass spec can be limited, and also the sensitivity. But the most striking disadvantage is that you have to deplete, fractionate, and digest your sample, and therefore completely change your sample for the analysis. And that leads to the problem when you want to translate mass spectrometry data into clinical use immunoassays. You have a high attrition rate of biomarkers or drug candidates, or you have a strong technical influence on your results. On the other hand, classical immunoassays use the native sample metrics, so you don't have any translational issues, but the problem is you have to select a few targets, so you can't do ELISAs for more than 1,000 targets. It would be possible, but yeah, it's quite time consuming and not easily doable. So the technique of Psyomics is in the middle. We cover more than 1,000 proteins on protein level and phosphorylation level. It's completely immuno-based. So we do not have the translation issue, but we have a large coverage and the sensitivity of classical immunoassays such as ELISAs. So we've combined the advantages of both worlds to really speed up discovery projects, research, everything. So as I said, we cover more than 1,000 proteins, and this enables a very comprehensive overview of changes within your biological system. So Our semi-targeted approach just enables faster research projects because you get really relevant targets out of the study for follow-up and as candidates for biomarkers or to evaluate your mode of action of a drug. Some platform features, the most striking advantage is that we can work with very low sample amounts and with various sample types. So we are not limited to any sample types. We can use plasma cells, tissue, spinal fluid, all on the same platform with adaptive protocols, of course. And the other advantage is that we can use very low amounts. So tissue, we can work with small biopsy samples with a few milligrams of tissue cells, up to or down to 200,000 cells, depending on the cell type, is feasible without any problems. And furthermore, we are not limited to human or mouse rat samples, but also large animal models are feasible to investigate on our platform. And as I mentioned earlier, the sensitivity of our platform is also in the range of classical ELISA assays. So all in all, you have a broad range of sample types with very low amounts and high sensitivity ranges. How does our platform work? It's pretty straightforward. We have a planar surface. We immobilize antibodies covalently, and then that's basically our technology. For the sample preparation, we isolate the proteins, label them with a fluorescent dye, and put them on the arrays. So that seems very straightforward. The details are a little more complicated, and it took about 10 years to figure out the optimal conditions. So, what does the array cover? We have a broad coverage of more than 1,000 proteins, as already mentioned, and we cover almost every protein class. The focus is on signaling, receptor transcription, but also structural proteins and a lot of proteins involved in immunity, immune cells, and that's especially relevant for the cancer field. In terms of pathways, we cover 135 pathways in total, but again, the focus was originally on oncology, in the oncology area, so therefore we have a lot of inflammatory pathways, a lot of immune cell related pathways, B cell, T cell, but also all the growth factor pathways, and lately we've added a lot of proteins covering neurological applications. So the platform has a broad range of applications, and I will talk a little more about this later on. Just to give you an example about our coverage, these are the pathways in cancer, and as you see, the green targets we cover with our array, and they are spread all over the pathway map. So you can derive changes in your biological system very easily and sort them according to pathway involvement, biological function, and also what's the meaning of the changes. So not only is one pathway changed, but you can say, okay, this increases apoptosis or proliferation, and that leads to a certain effect. So that's especially useful if you look for biomarkers in conjunction with biological mechanism. And that's especially useful if you want to patent your biomarkers, so you can strengthen the application with a biological mechanism. Another advantage of our platform is that we do not only look at the protein level, but also at certain post-translational modification levels, such as the phosphorylation, but others are under development, like ubiquitin, acetylation, and so on. And the striking advantage is that you get all the information of the protein level and the phosphorylation from the same assay. So you do not have to carry out several assays. You do not run into reproducible issues while carrying out different assays. And you also do not have the issues of combining data sets derived from different methods, assays, and so on. So it's a really speedy method to get a lot of information. Just to give you an example how it looks, on the left, you have a graph representing the 1,000 proteins on the array, and it's yeah, you have the protein abundance on the X and the phosphorylation abundance on the Y-axis, and you can just look which proteins change or have changes on phosphorylation level, on protein abundance, and then look into more details. That's depicted on the right. For certain proteins, just have a look at the details. So is the protein changed on phosphorylation level, on protein level, on both? And how do the sample groups behave? Are they homogeneous, heterogeneous? And this gives you a really nice feel of the data quality, the quality of the sample sets. Again, yeah, you get a lot of data out of the study, but also with our data analysis, a lot of information, so usable information, not just a data sheet with 500,000 to 1 million numbers. That's nice, it's a lot of data, but it doesn't give you anything to work with. So what we standardly provide is a study report, including comprehensive quality control of your sample sets, identifying outlier, subgrouping, confounding factors, all kinds of analysis, and that's also bespoken. So we will get in touch, discuss certain influencing factors with you, and then go on with the real analysis. And the study report furthermore contains differential proteins, differential phosphorylation, and then all the analysis that gives you really the information from that. So biological function of the proteins, protein interactions, affected pathways, and of course, we are always happy to discuss the implications of the basic data analysis and then take it a step further to really get what you want from the screen, so give you the maximum value from the study. So that was the overview of our platform, and before I go into the case studies, I would like to commence the Q&A session. So if you have any questions, now is the right time to get them out. Any questions?
Yeah, feel free to type your questions in the questions pane if you have any.
So I think if there are no questions, I've comprehensively explained why the platform is great. I will move on, but feel free to interrupt me anytime if you have some burning questions. But in the end, we will also have time for Q&A and really all your specific questions. I'm happy to answer them all. Okay, the first case study is from the checkpoint inhibitor field, so hot topic at the moment in the oncology area. And we conducted a study with the University Hospital here in Heidelberg and the German Cancer Research Center to answer the question, can we stratify melanoma patients that respond to an anti-PD-1 biologics therapy from patients who just don't? The question is clinically very relevant because the therapy is very expensive and does yield a lot of side effects. So it can be life-saving, but it can also be, yeah, just not doing anything for you except for the side effects. So the stratification is very important from various points of view. And the study conducted was from plasma samples taken prior to the therapy and after the second treatment cycle. So all in all, we identified more than 50 proteins able to distinguish responding from non-responding patients at baseline, and also proteins that were able to show the treatment success after the second dose, which is quite early, and would also mean a huge improvement for the clinical setting. So just two examples, protein A on the left side can distinguish between the non-responding patients and responding patients at baseline, so the two columns on the left, but doesn't change up on treatment dramatically, so it would just be a predictive marker. On the other hand, protein B would also be a predictive marker, but in the responding group, it has a huge increase in protein abundance and therefore could be considered a treatment response, and later on, if you see any changes back to the baseline level, it might also indicate treatment resistance. So that's very interesting protein, and we are currently in the process of setting up a larger study to follow all these results up. One example of the data analysis we could provide is look at the differential proteins, and we did this at baseline for the responding and non-responding patients. And just to sum it up really quickly, in the non-responding patients, we have an active immune system. So we have positive regulation of apoptosis, we have a lot of positive regulation of inflammatory response, and also a lot of proteins involved in the positive regulation of immune cell proliferation and migration. On the other hand, the responding patients have a negative regulation in the programmed cell death axis. So that's the PD-1, PD-L1 signaling axis, and PD-1 is the target of the biological drug. So that finding is expected, that we have a block which we could loosen by applying the anti-PD-1 antibody. But furthermore, we have a lot of regulation, so negative regulation for the immune response, for cell death in general, and also for inflammatory response. So these patients have a suppressed immune response to the tumor, and that's exactly what you expect to find, but we were kind of surprised to find it in this detail from plasma samples. Moving on to the next biomarker case study. It's the recurrence of bladder cancer, and this case study should give you a feel about what our data analysis can yield and what value a larger study with our platform can bring to your research projects. A little background about bladder cancer. So usually bladder cancer is diagnosed early. You have a non-invasive tumor which gets removed, but in about 60 to 70% of the patients, the tumor recurs within 5 years. So the question is, who will have a recurrence? And the answer is we don't know at the moment. So this creates the need for routine surveillance, creating high costs, high psychological burden for the patient, not knowing, do I have a recurrence or not? And paradoxically, this leads to a low compliance in the surveillance cytoscopy, so leading to an overall worse outcome for the patients which have a recurrence, because they go to the surveillance cytoscopy late, the tumor has regrown, it's invasive, and yeah, if you have an invasive tumor, your prognosis usually looks pretty bleak. So we wanted to find proteins that were able to distinguish responding recurrent from non-recurrent tumors and enable risk-adapted monitoring therapy and overall improve the outcome for all the patients. We found more than 200 proteins that were able to distinguish recurrent from non-recurrent tumors, and one was the active caspase 3 and 9. So what we did was look into the literature first, and we found several publications that correlated active caspase 3 levels, so increased levels, with worse prognosis for the patients. So that was in nice concurrence with our array data, and we found a lot of publications just showing exactly this fact. And upon conducting more literature research, we found that it was true in bladder cancer, but also in various other cancer entities like breast cancer. And there were several mechanisms postulated, and one was published in Nature Medicine that stated that increased apoptosis will lead to an increase in the inflammatory response, so creating a chronic inflammatory environment, which leads to the repopulation of the tumor and also increased angiogenesis of newly formed tumors. So this is just one example of a mechanism, but all in all, most of the papers postulate a chronic inflammatory environment as the main factor for the tumor regrowth. This is also nicely mirrored in our array data, so we find a lot of proteins that are involved in the inflammatory response as well. Looking not only at differential proteins, but just at different pathways, we choose the TGF-beta pathway as an example. So as I said, the platform covers 135 pathways, and we can have a detailed look at each and every one of the pathways. Just an example, TGF-beta. And in tumors with recurrence, the TGF-beta pathway is completely downregulated. So all the targets in green are downregulated in recurrent tumors, and the pathway inhibitor, the MAP kinase 3, is largely upregulated. So in tumors with recurrence, the TGF-beta pathway is basically knocked out, so it's not active anymore. Just one of the biological findings from the screen. And as I told you, the main goal was to find biomarkers, but at the same time, we can also look at biological mechanism behind the question, why does the tumor regrow? And we can also broaden it not only to single pathways, but also to biological functions. And as I stated, we have found a lot of active caspases, so 3 and 9, and this hints to more apoptosis. But on the other hand, we have also found that the calcium-induced cell death pathways are activated, and P53 as the guardian for the genomic stability is downregulated in tumors with recurrence. So we can look at a lot of biological functions, different implications for the system, and really get a comprehensive overview what is changed in the two settings, so recurrence and non-recurrence. Then we had an interesting question from a clinical collaboration partner stating that recurrent tumors are usually resistant to chemotherapy. And we thought, okay, that's an interesting finding, and let's see if we find proteins that are regulated that also hint in the direction or may explain why these tumors are resistant to chemotherapy. And we found a few. So we had an upregulation of NF-kappa B complex, so that mediates cell survival in general. We had the downregulation of the TGF-beta pathway, which I've already shown you, and that hints to less activity in apoptosis, and also the apoptosis inhibitor protein FLIP, which is also linked in literature with chemotherapy resistance, was upregulated in tumors with recurrence. So that gives you a really nice picture for a very specific question. Again, same question, chemotherapy resistance. We've looked a little further into the subject and also identified c-Jun as upregulated in our microarray screen. And c-Jun is also linked in the literature to chemotherapy resistance. And upon looking a little further, there are several studies confirming that upregulation of c-Jun on the transcription level in various cancer entities is always linked with worse survival and chemotherapy resistance. This was also published on the protein level, so immunohistochemistry. This is shown on the bottom of the slide, so that's taken from the literature, and on the top are our array data. So they fit really nicely to already published data, but give you a lot more. So you get new targets, new insights in the mechanism, and you get stratification biomarkers. So that's really all in one. You don't have to do several studies with several methods. Just use the SIO Discover. You get a comprehensive overview, you get biomarkers, mechanistic insight, and then go for the follow-up. Speaking of follow-up, usually when you go for biomarkers, mode of action identification, you do some kind of discovery study. you go into the literature, do a mass spec screen, do a lot of Western blots, or do a CytoDiscover study, and you identify a set of targets that are relevant and that you would like to use for a follow-up study. And this is something we can also offer with our platform. So the production of customized microarrays with your selected targets. Then we will talk about the antibody requirements. So what kind of quality, quantity, these are really important factors. And then we carry out the quality control in different steps. So there are various methods available. For example, the Western blot screen, which is not ideal for every antibody and every application. But also we have human protein arrays available with at least 9,500 human proteins, which gives you a really nice overview about the binding behavior of the antibodies and the specificity of your antibody. And this is especially important when you want to move into a diagnostic application, translational research, because when you build clinical decisions on your assay, you should be sure that your antibody is doing what it's supposed to do. And we enable really the comprehensive quality control that you can trust in the quality of the antibody. Having the antibodies, we will produce the arrays with our proven high quality protocols, do the quality control, and conduct a larger study with you. So going from the discovery stage to the verification stage, leading to a final biomarker signature, final stratification signature, so whatever the question you want to answer really is. Just an overview, what is Cytomics doing, where's the focus, in which areas can we work together. So cancer research, very general, everything with cancer. Our array was designed to profile different cancer entities, but we've taken it a little further, and one example is neurology. We have added a lot of targets for neurology and are moving into this direction a lot more lately. But also, if you're not in the cancer or the neurology field, our platform is really useful. For example, in the area of organ failure, aging research, toxicology screening, or an in-depth profiling of immune response, immune activation, or if you have more detailed research questions like transcription factor profiling. So we are really open for a large variety of questions, scientific areas. So just talk to us. I'm sure we will find something that fits your question and will hopefully deliver very good answers. That's the end. I hope I've not created more questions than answers, and I'm happy to answer any questions that may have come up during the presentation.
Great. Thanks, Ronnie. So there were indeed some questions that came in. If you're okay, then I'm going to start reading the first one to you.
Yes, please.
Okay. So one of the participants asked, what are the fold changes you observe in the examples you mentioned? For example, do you observe 20% increases or 6-fold changes?
The answer is yes. So in the studies we've conducted, we have always proteins that have really high fold changes, like 4, 6, but also we have proteins that were highly significant, but with low fold changes, like 0.5. But that really depends on your biological system, on your question, and how pronounced the changes really are. For example, when you go for a tumor profiling with a low number of actual tumor cells and a lot of stroma or other cells, you will get lower fold changes, but they are still significant. So yeah, the answer is probably really depends on your study, and we can observe large fold changes and very small ones.
Great. Another question here by the same participant, and I'll read this. According to your experience, what is the number of replicates that are necessary? Hold on. I mixed up a line here. I read it again. According to your experience, what is the number of replicates that are necessary to perform reliable statistical analysis from the raw data? Is the 3+1 rule something you follow, or do you feel confident enough to stick to 3 samples?
Good question. Question that comes up every time. So we would really divide this into the application and also the sample type. So if you go into cell culture analysis with a really defined system, a defined treatment, and defined conditions, 3 samples are okay. If you move to animal models with defined treatment or defined knockout models, 5 is okay, but there the variance is a lot higher compared to the cell culture. And if you go for treated animals, so various treatment groups, we would recommend to go for more replicates, as with every treatment and every group, you see an increase in biological variance that may overlay the actual variance of the treatment or of your knockout. And that's especially pronounced when you go into patient samples. So if you want to have biomarker discovery study in clinical samples from patients, we would say at least 10 replicates per sample group is the minimum where you get some significant results. And we would recommend to always go to the highest number of replicates that are feasible, because statistical significance is one thing, but also getting biological significance is the other thing. And we find that with more replicates, you get a clearer picture about the actual biological differences, which are not overlaid by individual characteristics of your system. Does that answer the question?
Well, we're going to have to see. I'm also a message to the audience. We will also answer those questions offline to you, and if there's any further comments, please also email us offline. So here's another question that came in. How can you analyze intracellular proteins? Do you have specific protocol for lysis of, e.g., blood cells to look at, for instance, phosphorylated proteins?
Very good question, and yes, we do. So we've developed special protein isolation buffers, special protocols for various types of cells and tissue. So, for example, when you go for a cytophosphoranalysis of a cell culture system, we would actually have a chat which cells, how are the cells treated, how are they cultured, and then give you specific recommendations how to prepare your cells prior to sending them to us. And then depending on the cell type, we have specialized protocols available. So for example, when you go for a liver cancer cell line, you treat it differently compared to a kidney cancer or some kind of blood-borne cancer cells. So that kind of depends what system you have, but we have conducted a large number of studies and have really adapted protocols for many sample types. And if you have something really new, which we've never seen before, we are always happy to conduct a trial feasibility study with you to find the optimal conditions and then move into the real study.
Great, thanks, Ronnie. So I'm going to read the last question to you. It's more of a general question. How good is the reproducibility and the sensitivity of the platform in comparison to mass spectrometry?
So both is better. Now, depending actually on this setup of the mass spec, if you go for a shotgun approach, our sensitivity and reproducibility is better, definitely better. So the mass spec has several problems. One is the method itself. It has roughly coefficients of variations of 20%, and then you have the data acquisition and data analysis. So that can introduce a large bias, and usually if you go for a data independent analysis, you have a CV of another 20 to 50%, And if you go for data dependent analysis, you reduce this to about 10 to 20%. So overall mass spec, let's say it's roughly 50% CV between analysis to analysis, and we are consistently below 10%, and during the last studies we actually reached values of about 5% for many sample types. So that's the answer to the reproducibility question. And sensitivity, as I mentioned, we are in the range of ELISA assays, and that means femto to picomolar range. And in the mass spec shotgun approach, you're usually factor 10 to 50 above that. So our platform is a lot more sensitive than the general look at everything mass spec approach. Of course, when you go for other approaches like the multiple reaction monitoring, the sensitivity of the mass spec can increase dramatically and reach the levels of classical immunoassays. But that's a lot of work to establish. It takes a lot longer, and you kind of limit your samples, so the set of proteins you look at, to very few, and then the largest advantage of mass spec, looking at many targets in parallel, just vanishes again.
Great. Thank you, Ronnie. So I see there are no further questions at this point. So I want to thank everyone for attending this webinar presentation. We also want to thank our speaker. We will make the presentation slides and the webinar file available on our website. If you have any questions, please contact also the Sciomics team directly. Also feel free to check out our website for updates, future events, and webinar announcements. So I will conclude this webinar now. Thank you again to everyone for dialing in. Goodbye.